long before the mountains were carved into borders, before languages gave names to rivers, and before kings etched their reigns in stone. There were people, silent, unnamed, and sowing seeds into earth that would remember their footsteps longer than any empire. Their bones are buried beneath the dust of Balkh, Samangan, and Helmand. Their faces are lost, but their legacy survives in the genomes of modern Afghans. These weren't conquerors. They were the first builders, farmers, mothers, elders. They domesticated wheat before war was born. They tamed sheep before chariots thundered across the plains. And in their quiet revolutions, planting, weaving, tending, they planted something else as well, a genetic heritage that still pulses in the blood of millions. Science calls it the Neolithic, but it was more than a time. It was the dawn of identity. Recent genomic studies, scanning through the DNA of Pashtun, Tajiks, Uzbeks, and Hazaras, uncover a truth so buried, so unspoken, that even national histories seem to have forgotten it. Despite all their ethnic divisions, political disputes, and cultural schisms, these peoples descend from the same ancestral population, one that began to take shape nearly 10,000 years ago. This early population was likely formed from the mixing of Iranian plateau farmers with a smaller thread of Anatolian ancestry. They carried the genetic footprints of J2, LM20, and the ancestors of R1A. They lived off river valleys and trade paths that hadn't yet become war routes, and they spread slowly across a land that didn't yet know its own name. We know this because of the genetic homogeneity visible across all Afghan ethnic groups at the base of their family trees. The Pashtun, often mythologized as Aryan warriors, share a genetic ancestor with the Tajik stonemason. The Hazara boy in Bamyan shares lineage with the Uzbek grandmother in Shebergan. It is not folklore. It is scientifically observed kinship, traced through genomic data, autosomal similarities, and unbroken strands of mitochondrial DNA that link today's children to Neolithic mothers, whose graves have long been erased. And then came the shift. Around 4,000 years ago, the Bronze Age arrived like a thunderclap. Urban settlements emerged. Metallurgy, organized religion, and trade exploded. In what is now northern Afghanistan, the Bactria, Margiana archaeological complex rose from the earth like a forgotten Atlantis. Cities with walls, temples, storage granaries, even early forms of writing. And with them came a subtle shift in genes. Localized differentiation began. Isolations, migrations, intermixing with new waves of travelers and settlers, perhaps some from the south, carrying Indus Valley blood, perhaps others from the west, bearing Caucasian ties. Afghan DNA began to branch. Haplogroups diverged. But through it all, the seed of Neolithic ancestry remained untouched at the root. The Hindu Kush stood as both witness and architect. Its jagged ridges acted as a sieve, letting some genes through, blocking others. Northern populations, surrounded by mountains and limited interaction, retained a more conserved gene pool. Southern regions, connected to the Indian subcontinent, absorbed more diversity through what would become the arteries of the Silk Road. And yet, amidst this growing complexity, one truth endures. The Afghan genome is not a mosaic of alien fragments. It is a tapestry woven from a single, ancient thread, frayed at the edges, yes, but unbroken at its core. The farmers who once tilled the land beneath Balkh may have no statues, no surviving names, but their DNA still breathes inside every Afghan heart that beats today. When the wind began to howl across the open steppes of Central Asia, it didn't just carry dust. It carried a legacy, one that would etch itself into the bones of poets, prophets, kings, and tribes yet unborn. A legacy called R1A, a haplogroup that would come to define the Indo-European soul. 3,500 years before the birth of written Persian, long before Pashto echoed through the valleys of Nangarhar, a wave of horse-bound migrants surged across the grasslands of the Pontic-Caspian steppe. They carried fire, metal, cattle, and within them, a silent genetic code. That code, R1A1A198, would reshape the paternal lines of South and Central Asia forever. They were not the first to ride, but they were the first whose ride rewrote language, ritual, and kinship. From their tongues emerged the ancestors of Sanskrit and Avestan. From their rituals rose sky gods and sacred fires. But deeper than their myths was their blood, and it flowed, silently, into what is now Afghanistan. Among Pashtun today, this lineage doesn't whisper. It roars. Over half of all Pashtun men carry R1A, making them one of the densest concentrations of this haplogroup on Earth. But its presence is not uniform. In the Tajiks, it appears with slightly less frequency, tempered by greater paternal variety. Still, its echo is clear. The gene remembers, even when the borders, tribes, and empires forget. These weren't mass invaders. They were slow settlers, moving in waves, not storms. Archaeogenetic evidence suggests that the steppe influence in Afghan DNA didn't arrive all at once, but through centuries of intermarriage, pastoral exchange, and tribal absorption. They didn't wipe out those who came before. They mixed with them. And in that mixture, a new cultural and genetic hybrid was born. The timing of their arrival aligns with the twilight of the Bactria, Margiana complex. As Bronze Age cities waned, these newcomers brought with them fresh technologies, mobile economies, and the Proto-Indo-Iranian worldview. The sacred texts of the Rigveda and the Avesta bear the stamp of steppe cosmology, chariots, solar deities, fire cults. But it is the DNA that confirms what linguistics could only guess. The R1A subclade found in Afghans, specifically R1A-Z93, links them directly to the eastern branches of Indo-Iranian migrations. This is the same lineage found among Brahmins in northern India, Scythians of the Eurasian steppe, and even ancient graves in the Altai Mountains. In essence, it is a pan-Indo-Iranic marker, a shared genetic thread running from Tehran to the Ganges. But here's the mystery. While Pashtun hold high R1A frequencies, they also possess lineages like G2C, M377, nearly absent in other groups. This rare branch suggests that even within steppe descent, there were layers, perhaps different tribes, different waves, different dialects of the same wind. And then there's the cultural memory. Many Pashtun tribes claim descent from ancient Aryans. Some even hold myths of descent from the lost 10 tribes of Israel or soldiers of Alexander. But while those stories shimmer at the edge of folklore, R1A offers something more concrete. It offers roots that predate empires, roots buried not in scripture or song, but in the double helix of life itself. For the Tajiks, the R1A story is more muted, but not absent. Their greater paternal diversity speaks to a people who have long stood at crossroads, absorbing, translating, and bridging cultures. Their DNA reflects not a singular storm, but a mosaic of breezes from Iran, Central Asia, and the distant steppes beyond the Oxus. As the Indo-Iranian speakers spread, they seeded not only language, but also a new structure of society. Caste, kinship, hierarchy, all intertwined with paternal descent. And R1A became not just a gene, but a silent social architecture, one passed from father to son, across centuries of upheaval and resilience. But the gene carries no pride. It remembers no conquest. It is we who attach meaning to it. Today, Afghan men who carry R1A may never know the names of their ancestors who rode beneath a sky without borders, but their blood speaks a language older than empire, one that reminds us, in every village, in every tribe, there might still live a whisper of that ancient migration. Not foreign, not distant, but utterly human. There's a face in the highlands of central Afghanistan that tells two stories. One is written in cheekbones curved like the edge of the Altai. The other whispers through skin tone, in eyes dark with memory. These faces belong to the Hazara, a people suspended in genetic contradiction, standing at the crossroads of history's most brutal collision. Their language echoes Persian. Their culture breathes the soil of the Hindu Kush. But inside their cells, in the unbroken thread passed from father to son, a different origin pulses. Geneticists call it C3, M217. Historians call it the Genghis Khan signature. And nearly 40% of Hazara men carry it. This isn't legend. It's molecular testimony. The same Y chromosome lineage has been identified in populations stretching from Mongolia to Manchuria, in families that trace their paternal ancestry back to the 13th century. And now, deep in the valleys of Bamyan and Daikundi, it emerges again, like a ghost that never left. But the Hazara genome isn't just a relic of conquest. It's an archive of resistance, survival, and silent intermingling. Alongside their Mongolic paternal legacy, Hazara maternal DNA reveals a profound mixture. Approximately 35% of their mitochondrial lineages trace to East Asia, haplogroup C-D-F, while the rest align with West Eurasian ancestries. This genetic split mirrors the structure of an invasion. Male soldiers arrived. Local women remained. Together, they created a new people whose identity was forged not in diplomacy, but aftermath. Still, some fragments defy the story. In several Hazara lineages, scientists have discovered haplogroups of African origin, such as BM60, and Levantine markers like E1B1B1C1M34, commonly associated with Jewish and Middle Eastern populations. These anomalies don't follow a pattern. They appear like static in an otherwise decipherable signal. Some researchers suggest ancient trade routes or coerced migrations may explain them. Others admit we simply don't know. What we do know is this. Hazara DNA is unlike any other in Afghanistan. Where Pashtuns show concentrated paternal ancestry and Tajiks reflect Iranic continuity, the Hazara genome is a fusion, a frontier, the visible evidence of a moment when the Mongol Empire didn't just conquer cities, it altered the future. But their distinctiveness isn't only genetic. It is geographical, too. The Hazara live in highland isolation, in regions historically marginalized and often excluded from central authority. This separation, enforced over centuries, may have helped preserve their East Asian genetic components. Isolation became a vessel, containing not just a people, but their molecular past. And then there's the silence. Unlike empires that leave behind monuments, Hazara history survives through whisper and inheritance. Their oral traditions speak of Genghis Khan. Their legends recall horsemen from the north. But the DNA carries what the memory cannot. It tells us of women whose names were never recorded, of children born between siege and surrender, of lineages created in the shadow of empire. Hazara faces today carry these shadows. They carry also something deeper, a record of how identity can emerge from trauma, how culture can take root in collision, and how blood can bind the conqueror and the conquered into one. They are not simply Mongol descendants. They are something else, something history struggles to name. To understand the Hazara is to read the genome like a palimpsest, layers overwritten, never fully erased. A people born of rupture, whose DNA speaks not of belonging or purity, but of survival through fracture. The Hindu Kush doesn't ask permission to divide. It simply rises, jagged, cold, and absolute. A wall of stone that split the sky long before it split civilizations. And in its silence, it shaped the genetic fate of millions. Afghanistan's mountains aren't just geography. They're destiny. Where rivers couldn't reach, neither could people. Where elevation broke the earth, it broke bloodlines too. The genome carries the map of these ridges, not in contour lines, but in lineage frequencies, bottlenecks, and ancient silences. In northern Afghanistan, hemmed in by the taller ranges, something peculiar happened. The genetic diversity began to narrow. Fewer Y-chromosomal lineages persisted. In contrast, the southern regions, more exposed to the plains of South Asia, saw influxes, admixture, and a larger variety of paternal markers. The difference wasn't just numerical. It was structural. Isolation carved pockets of conserved ancestry, while connection fed complexity. But not all boundaries divide. Some corridors, like the routes that threaded through Bamyan, Herat, and Kandahar, became arteries of exchange. For centuries, caravans crossed these paths, laden with silk, lapis, spices, and silently, with genes. What we call the Silk Road wasn't just a trade network. It was a genetic highway. Each merchant, each nomadic tribe, each lost caravan left behind traces more permanent than footprints. Genetic studies now trace shared markers between Afghans and populations as far as Iran, Pakistan, India, and even Central Asia. These aren't coincidences. They are the molecular shadows of routes once alive with conversation, commerce, and cultural collision. In places like Herat, the genome tilts westward, showing affinities with Iranian populations. Toward Jalalabad, the tilt turns east, aligning with South Asian gene flow. Mazar-e-Sharif and Sheberghan, positioned near historic Oxus crossings, show greater Central Asian input, particularly from Turkic and Uralic ancestries. But what's perhaps more remarkable is that these variations aren't confined to ethnic boundaries. They cut across language, tribe, and religion. Geography has more authority than ideology. A man in Faryab and a man in Khost may speak different tongues, pray in different ways, and trace their cultural traditions through entirely separate narratives. But their DNA may share a grandfather, separated only by the arc of a mountain range. Scientific analysis backs this with high-resolution data. Regions cut off by geography exhibit founder effects, where rare lineages become common, simply because no new ones enter. Meanwhile, trade cities, with centuries of incoming populations, show tangled genetic webs that defy simple classification. Even the concept of ethnic purity dissolves here. When the mountains protect, they also entomb. And when the roads open, they rewrite. One striking example lies in the Nuristani population. Tucked away in eastern valleys, long thought to be isolated, they show surprising haplogroup continuity with Indo-Aryan groups, suggesting preservation, not interruption, of ancient descent. Meanwhile, Turkmen and Uzbek populations in the north display lineages like Q1A2 and C3, inherited from waves of steppe migrants who entered through open corridors. And yet, these migrations were never unidirectional. Afghan genetic history is filled with backflow. People who entered, settled, blended, and whose descendants turned back eastward, westward, or southward. In genetic principal component plots, Afghans don't fall neatly into one cluster. They float in between, a genetic spectrum suspended between Iran, India, and the steppe. This floating isn't confusion. It's evidence of ancient interconnectedness, forged in dust and altitude. In the end, the mountains gave Afghanistan something no empire could, a genetic fingerprint that resists homogeneity, a landscape that built complexity not through war or conquest, but through the slow, grinding pressure of time, stone, and silence. In the clean geometry of population genetics, patterns are everything. Ancestry moves in predictable waves. Markers emerge where they should, follow geography, align with language, and bow to history. But sometimes they don't. Sometimes, a sequence appears where it has no right to be. A haplogroup surfaces in a mountain village that's never seen its kind. A genetic ghost whispers from inside a population that carries no memory of it. And in Afghanistan, these whispers are louder than anywhere else. Among Hazara lineages, already complex with East and West Eurasian roots, researchers stumbled upon something startling. A small cluster of men carrying haplogroup BM60, a paternal line common in parts of sub-Saharan Africa. But there is no historical record of large-scale African migration to central Afghanistan. No known trade corridor, no political alliance, no enslaved population that explains it fully. And yet it's there, embedded in the Y-DNA of a people who trace their identity to Mongol conquests and Persian language. The marker appears like a memory misplaced, as if someone rewrote a page of history and never told the rest of the world. It's not alone. In several individuals across different Afghan groups, from Uzbeks to Hazaras, scientists have discovered traces of haplogroup E1B1B1C1M34. This lineage, commonly found in Jewish and Levantine populations, carries echoes of Semitic heritage. Yet in Afghanistan, it hides in fragments, scattered, unanchored. Was it carried east by early merchants? By converts during the Khazar era? Or did it arrive through forced movement, trade, or something else we've lost to time? Then there's the presence of Melanesian mitochondrial DNA, a maternal line associated with populations in Papua New Guinea and surrounding islands. In the narrow alleys of Kabul or the valleys of Daykundi, this seems impossible. Melanesia lies thousands of kilometers away, across oceans and empires. And yet, in rare Afghan samples, the maternal signature appears, like a forgotten tide that once washed ashore and left a single footprint in the sand. These anomalies defy the clean arcs of migration. They challenge the tidy story of Indo-Europeans, Mongols, Turks, and Persians. They suggest that somewhere in Afghanistan's buried past, encounters occurred that no one recorded. Encounters outside the maps, beyond the scrolls, before the historians ever thought to look. Some of these genetic fragments may be the result of ancient admixture events, long before Afghanistan bore that name. Others may stem from individual episodes, a single traveler, a family displaced, a child born from a brief intersection of worlds. In genetics, even one person can plant a legacy. These lineages don't change the dominant narrative, they deepen it. Afghan DNA isn't random. It's layered. It contains the expected foundations: Neolithic farmers, Indo-Iranian speakers, steppe riders, Mongol invaders. But between these dominant threads lie filaments of mystery, tiny anomalies that don't belong, and yet do. These aren't just scientific curiosities. They're personal. They tell us that no history is entirely known, that sometimes the rarest line in the genome carries the most ancient story, and that every anomaly, every misfit sequence, is a clue, not a flaw. In a world obsessed with belonging, these ghosts remind us that identity isn't about purity, it's about complexity. That who we are isn't determined by dominant genes, but by the forgotten ones, the ones that survived not because they conquered, but because they endured. There's a cost to legacy when tradition turns inward. In the highlands and valleys of Afghanistan, families have survived centuries of war, empire, and collapse, held together by kinship, language, and blood. But in that closeness lies a quiet danger, one not born of swords or politics, but of chromosomes. Across the country, nearly half of all marriages take place within the family, between cousins, uncles, and nieces, sometimes double first cousins. In a culture where lineage is protection, where tribal lines are tightly drawn and outsiders are risk, endogamy becomes not just normal, it becomes expected. But genes are indifferent to customs. Hidden within every human genome are silent mutations, most of them harmless, lying dormant. But when two closely related individuals reproduce, those dormant traits can awaken. And in Afghanistan, they have. Recent studies show that nearly 75% of known genetic disorders in Afghan populations are autosomal recessive, inherited only when both parents carry the same faulty gene. Among infants, these disorders are not rare events. They are an epidemic. Metabolic syndromes, neurological defects, chromosomal instabilities, conditions that in many parts of the world are statistically improbable, here become common. In children under the age of 2, the rate of inborn errors of metabolism nears 39%. That's not just a number. That's a generation born into biochemical malfunction, often without diagnosis, without treatment, without a name. The numbers don't just speak. They scream. 1 in every 784 births here is marked by a genetic error, so severe it alters the body's ability to process life itself. These aren't rare, exotic mutations from a distant land. These are inherited legacies, passed quietly from cousin to cousin, from parent to child, over generations of well-meaning, culturally sanctioned choices. And yet, the tragedy deepens. Afghanistan lacks widespread genetic screening. Many families never know they carry risk. Laboratories for diagnosis are scarce. Specialists rarer still. In remote districts, a child's seizure, blindness, or paralysis is often blamed on fate or misfortune, not a strand of DNA that could have been anticipated, perhaps even prevented. These disorders do not distinguish between ethnic lines. Pashtun, Tajiks, Hazaras, Uzbeks, all are affected, because the problem is not tribal. It is structural, cultural, and heartbreakingly, generational. Some chromosomes are struck more than others. Studies show recurring mutations on chromosome 6, a hotspot tied to immune regulation and metabolic function. Chromosomes 11 and 12 follow close behind. In contrast, no documented mutations have yet been observed on chromosome 4, 19, or the Y chromosome in affected Afghan samples. A silence that itself tells a story about which regions of the genome are absorbing the burden of this crisis. There is no blame in the double helix, only consequence. Many families marry within because they must, because survival depends on trust, because centuries of conflict have narrowed the lines of safety and belonging. But genetics does not yield to war or poverty. It only amplifies what's given to it. And so, in a country that carries within its DNA the story of ancient civilizations, of Indo-Iranian migrations, of Mongol conquests and Silk Road crossings, a new chapter unfolds. One not of conquest, but of collapse. A collapse not of identity, but of cellular function. The burden of heritage here is not abstract. It cries in hospital rooms, in homes without doctors, in mothers who bury 3 children before they understand why. But knowledge is power. The very same science that mapped Afghan ancestry can now illuminate its future. With genetic counseling, expanded diagnostic services, and community awareness, this cycle can begin to shift. Slowly, carefully, humanely, because in every child lies not just the memory of the past, but the potential to rewrite what comes next. In the swirling dust of Central Asia, between collapsing dynasties and forgotten roads, lies a secret the world almost missed. It's not in the ruins, not in the manuscripts, not even in the legends. It's inside the cells, unspoken, microscopic, and eternal. Afghan DNA is not a singular lineage. It's not a tale of origin. It is the biological archive of civilization's collisions. When we sequence genomes from Herat to Jalalabad, we're not just studying genes. We're opening time capsules. Afghan DNA doesn't speak in straight lines. It speaks in echoes, the kind that bounce off empires, twist through invasions, and return carrying traces of strangers turned ancestors. This is not the story of a race. It's the story of how race dissolves when history passes through bodies. Pashtun carry Indo-Iranian codes layered with local adaptations. Hazaras preserve Mongol imprints entangled with Central Asian and West Eurasian threads. Tajiks and Uzbeks act as bridges, not metaphorically, but genetically. The people of this land don't sit at the borders of Asia. They are the overlap, the point of convergence, the human Venn diagram. In a world obsessed with dividing cultures by blood, Afghan DNA exposes the illusion. It shows us that languages shift, empires fall, religions change, but the genome remains loyal to what came before. And what came before was interconnectedness. Even when walls rose, the kind made of stone or belief, genes moved freely. Through marriage, through trade, through survival. The body always knew what the map forgot, that no one lineage stays untouched. That's what makes Afghan DNA so important beyond Afghanistan. It teaches us that identity is not possession. It's process. It teaches us that ancestry isn't fixed. It flows, like rivers through generations, sometimes drying, sometimes flooding, but always moving forward. And it teaches us that every person on Earth carries pieces of lost stories, forgotten migrations, buried alliances. In Afghanistan, those fragments survived the burning of libraries, the crumbling of temples, the silence of exile. Because they weren't written in books, they were written in blood. Modern science now has the tools to read those pages. Principal component analyses, haplogroup breakdowns, autosomal plots. They're not just academic data points. They are mirrors, showing humanity to itself without the veil of politics or power. In these mirrors, we see that every Afghan is the child of farmers from the Fertile Crescent, warriors from the steppes, nomads from Inner Asia, traders from Persia, settlers from the industry. And yet, they are not diluted. They are defined not by purity, but by resilience, by the ability to absorb trauma and still transmit memory, by the survival of structure in the chaos of history, by the fact that after thousands of years of conquest, isolation, intermarriage, and collapse, there is still a people who carry within them the most complete genetic story of Asia. This is what Afghan DNA teaches the world. That complexity is not weakness. It is strength. That diversity is not decay. It is legacy. And that the body, when left to tell its story, speaks a truth older than myth and deeper than nation. In every Afghan genome, there is proof that humanity does not move in straight lines. It weaves. It bleeds. It blends. It endures. And in doing so, it remembers, even when we forget.